Have you ever wondered how plants make their own food? I guess by now, since we're all in 12th grade, we are somewhat familiar with this process. Yes, it is called photosynthesis, an amazing process by which plants produce their own food. Good day, everyone. I'm Hannah Sophia from St. Ignatius, and I'm excited to share with you how this incredible process works and why it's so important for life on Earth. When we say photosynthesis, it is not just plants that can undergo this process. Some protists and some bacteria can do too. However, for this lesson, we will just focus on how plants undergo this process. Let us start by understanding why photosynthesis is so important, not just for plants, but for all life on Earth. Photosynthesis matters because it produces oxygen, a gas that we absolutely need to breathe. Plants are major producers in ecosystems, forming the base of food webs. When we say plants need to produce their own food, it talks about a sugar that they need, specifically glucose. Us humans need glucose too, but we get it from what we eat. Plants, however, get to make their own glucose in a process called photosynthesis. Now let's take a look at the overall photosynthesis equation and compare it to cellular respiration, which is how cells get energy. Here is the balance equation for photosynthesis. Notice it shares components with cellular respiration, the process that produces ATP, or as we all know, energy, in cells. We can see how the inputs in photosynthesis can also be seen in the outputs for cellular respiration, as well as the inputs for cellular respiration can be seen in the outputs of photosynthesis. While the equations are not exactly reversed, they involve the same molecules in a cycle. To capture light for photosynthesis, plants rely on specialized pigments. These specialized pigments is called chlorophyll. Chlorophyll is a pigment that can be found in the chloroplasts of plant cells. Chlorophyll absorbs mostly red and blue light but reflects green, which makes plants appear green to our eyes. Let us recall that visible light has different wavelengths, and those different wavelengths of light have different colors, which explains why green is not the only color we see in plants. There are two major reactions that occur in the chloroplast that together make up photosynthesis: the light-dependent reactions and the light-independent reactions, or also known as the Calvin cycle. Let's explore how these work.
Good day, everyone. My name is Krisa May Itanjo, and I will start by introducing the light-dependent reaction, the first stage of photosynthesis. This stage happens inside the chloroplast, the organelle and plant cells, where photosynthesis occurs. Amo ini ang lugar na mo-capture sa sunlight, specifically the reaction takes place sa thylakoid membranes. These talk structures inside the chloroplast where sunlight is absorbed and converted into energy. So the first important structures here is a photosystem 2, a protein complex that absorbs sunlight. The pigment chlorophyll absorbs the light or adlaw, which excites its electrons. Murag gi-charge sila by the sun. These excited electrons are now ready to move along the electron transport chain. And for the next steps, including splitting of water, oxygen release, and production of ATP and NADPH, I will now pass it to my groupmate, Kimberly Dugmukan.
Now that my groupmate Krisa already tackled about the basics of this topic, and for you to have a better understanding and have enough knowledge of what I'm going to discuss next, let us now move on and continue the discussion of light-dependent reaction. As Krisa mentioned earlier, the light-dependent reactions occur inside of the thylakoids within the chloroplast. Now what happens in light-dependent reactions? Let me read this to you. This is the first stage of photosynthesis where light energy is converted into chemical energy in the form of ATP and NADPH. It takes place in the thylakoid membranes, which was mentioned earlier, and uses light energy to split water, produce ATP and NADPH, and releases oxygen. Now let me take you step by step through the process of light-dependent reactions. Now going to the thylakoid, which where LDR occur, and Focusing on the thylakoid membrane, this is where light particle will strike photosystem 2 and pigments such as chlorophyll are found and is where light gets absorbed. In photosystem 2, it is a protein pigment complex in the thylakoid membrane containing chlorophyll 680 nanometers. It absorbs light energy and excites electrons to a higher energy state. When this happens, electrons in chlorophyll gain energy or get excited. This process is what we call photoactivation. Photoactivation is the process where light energy excites electrons in chlorophyll, as what I've mentioned earlier. It will allow them to move to a higher energy level and begin the flow of the electrons. These excited electrons leave chlorophyll inside the photosystem 2 and move to something called an electron transport chain. The electron transport chain is a series of protein complexes and electron carriers such as plastoquinone, cytochrome complex, and phytocyanin. It is in the thylakoid membrane. It also transfers excited electrons from photosystem 2 to photosystem 1. Now, because chlorophyll lost electrons, it needs to replenish or replace those lost electrons. So it's going to take the electrons from water. So basically, to replace them, water is split and its electrons go to chlorophyll. This process is called photolysis. This is the process of splitting water molecules using light energy in photosystem 2. It produces oxygen gas as a byproduct released in the atmosphere. It also releases protons that contribute to a proton gradient. So when water is split, electrons, oxygen, and hydrogen ions are produced. This is where most of us know the oxygen made from the process of photosynthesis came from of plants. And in this process, it acts as the byproduct, as what I've said earlier, or a waste product released in the atmosphere. Now going back to the electron transport chain, plastoquinone is going to carry the electrons to cytochrome complex. And as the electrons pass through that complex, what's going to happen next is it's going to pump protons from the stroma that is from outside the thylakoid into the lumen or the inner thylakoid space. So protons concentration in the stroma will decrease, while proton concentration inside the thylakoid will going to increase. So as this happens, there will be a buildup of hydrogen ions inside the thylakoid space, or what we call the lumen. And there's a very little hydrogen ions in the stroma or outside the thylakoid. Then what's gonna happen is, these hydrogen ions, due to this concentration gradient, they will begin to flow through an enzyme called ATP synthase. ATP synthase is an enzyme complex embedded in thylakoid membrane. It uses energy from proton flow to catalyze synthesis of ATP. It also produces ATP, the energy currency needed for the Calvin cycle. Now, the process of proton flow is what we call the chemiosmosis. To further elaborate this, let me read this to you. It is the process where protons move down their concentration gradient through the ATP synthase enzyme channel in the thylakoid membrane. The flow of protons through ATP synthase provides the energy required for the enzyme to convert ADP, an inorganic phosphate, into ATP. As those hydrogen ions flow through that enzyme, this protein will rotate in such a way that it's going to combine ADP and phosphate to make ATP, which is a key product of the light-dependent reaction. When the electrons reach the end of this first electron transport chain, they go to photosystem 1. Photosystem 1 is a protein pigment complex with chlorophyll absorbing light at 700 nanometers. Reabsorbs light energy to re-excite electrons received from ETC, or the electron transport chain. It transfers high-energy electrons to ferredoxin, an electron carrier. So going back to the process, in photosystem 1, it is where the light excites them once again. They travel down a second, shorter electron transport chain where they are accepted by a molecule called NADP+. When it accepts the electrons, it also accepts hydrogen and becomes NADPH. NADPH is also an electron carrier and another key product of the light-dependent reaction. ATP and NADPH are the critical products of the light-dependent reactions that are needed to the next reaction, which is the Calvin cycle, which will be discussed by our groupmates Mel and Clark.
Good day everyone. So now let's proceed to the process of the light-independent reaction, or what we call the Calvin cycle. So first, let's now know its definitions. So it is the second stage of photosynthesis, which was discussed earlier on photosynthesis. Second, it happens in the stroma of the chloroplast, the fluid-filled space around the thylakoids. So, amin siya. Ayan, amin siya. Unlike the light reactions, it doesn't need light directly, but it depends on the product of the light reaction, which is the ATP and NADPH, to work. So, ang ATP, amin siya ang like, let's say like, battery yun siya, energy yun siya, let's say like battery yun siya. And NADPH is the carrier of the electrons. So, its main job is to use CO2, or what we call carbon dioxide, and energy, which is the ATP and NADPH, to make glucose or sugar. So, yeah. So, let's proceed to the three main phases of the Calvin cycle. So, first is the carbon fixation. So, CO2 enters, or the carbon dioxide enters the cycle from the air. An enzyme called rubisco attaches CO2, or the carbon dioxide, to a 5-carbon molecule called the RUBP, or ribulose biphosphate. So, ako na simply explain this. Okay. Lang ta lamang, as kuan is gibreathe ng plant ang carbon dioxide. So, pag-breathe ng plant sa carbon dioxide, and helper enzyme, which is what we call rubisco, is mo-attach siya carbon dioxide. And then, pag-attach niya is mo-tibo dayon siya into a 5-carbon molecule, which we call the RUBP, or the ribulose biphosphate. Okay, this makes an unstable 6-carbon compound which immediately splits into 2 molecules of 3-PGA, which is the 3-phosphoglycerate, each with 3 carbons. So, kay tungod kay attach niya sila into 3 carbon is unstable siya. And then, mabungkag siya into what lang ko na 3-PGA, which is the 3-phosphoglycerate. So now let's proceed to the second phase, which is what we call the reduction. So the reduction, or the energy input. So the light gives us reaction ATP and NADPH. Now they are used here. ATP adds energy and NADPH donates electrons to 3-PGA. This changes 3-PGA into a high energy carbon sugar called G3P, or the glyceraldehyde 3-phosphate. So explain ko yun ng more simpler. So, let's say for example, the plant already made energy from the sunlight. Now, it now uses this energy to change the carbon dioxide into sugar pieces. So, gihimo na ng sugar pieces ang carbon dioxide. So, the new sugar piece, it is what we call the G3P, which is rich in energy. Ano yun siya? So, gihimo lang niya ang carbon dioxide into a sugar piece, which has a lot of energy, rich in energy siya na sugar piece.
Good day everyone. I am now going to discuss the third phase of the light-independent reaction, also known as the Calvin cycle. This phase is called the regeneration. So most of the G3P is not used to make glucose yet. Instead, ATP is used to rearrange some G3P molecule back into RUBP, the molecule we started with. This allows the cycle to keep going and accept new CO2. After carbon is fixed and reduced into G3P, or glyceraldehyde 3-phosphate, what happens next? That's where regeneration comes in. The regeneration phase converts 5 molecules of G3P back into 3 molecules of RUBP, the cycle starting point, so the process can continue. This conversion requires ATP, around 3 ATP molecules per regeneration cycle. Importantly, only 1 G3P molecule exits the Calvin cycle each time, to be used in sugar or carbohydrate biosynthesis, while the rest supports the cycle continuation. Now let's look at the outputs of the whole cycle. For every 3 CO2 molecules that enters the cycle, 6 G3P molecules are made. Out of these, 1 G3P exits the cycle to help build glucose and other sugars. The other 5 G3Ps are used to regenerate RUBP. This means that the cycle has to turn twice, using 6 CO2 molecules, 18 ATP, and 12 NADPH to produce 1 full glucose molecule. To wrap up, regeneration phase is vital for the Calvin cycle. Continuity ensures RUBP is replenished while yielding the G3P building blocks for sugars and starch. Without it, carbon fixation would grind to halt. That's all. Thank you.